Today we're going to learn how to make a pivot table in Excel using our yeast phenotype data. The purpose of using a pivot table is to help us summarize and explore the data more easily. Instead of scanning through rows manually, a pivot table can instantly calculate totals, averages, or group comparisons, helping us spot patterns and trends in the results. If you haven't already, please download the Excel sheet from the Canvas link so you can follow along. Your spreadsheet should look something like this, with multiple columns as well as multiple data points. For this tutorial, I'm going to use a random subset of the full dataset to keep things simple and easy to follow. Let's take a moment to explore the data. In the first row, we have our column headers. These are the labels that describe what each column represents. We have section number, group number, transformation type, nanograms of plasmid added to the transformation, nanograms of HDR added to transformation, number of white colonies, number of red colonies, total number colonies. These headers help us organize and structure the data so we can analyze it in a meaningful way using our pivot table. Now let's talk about something that comes up a lot when cleaning CRISPR experiment data. What do you do if one of your plates has 0 colonies? If the plate was set up correctly, same cells, same volume, and there's no obvious mistake, then keep that data point. It might actually reflect some real variability, especially if you saw growth in your other plates. But if you know there was a technical issue, like forgetting to add DNA or the plate was contaminated, then it's okay to remove that data point. The best way to check is by looking at our comments section and seeing what could have potentially went wrong with the experiment. We have filters so we can quickly scan for any plates that had 0 colonies. First, go up to the top menu bar, click on Data, then select Filter. Now you'll see that filter buttons have been added to each column. Scroll down to the total number colonies and click on the drop-down menu. A new window will pop up and we will deselect Select All. Go ahead and click on 0. With our filter on, now we can see all the groups that received 0 colonies. We're also going to set another filter, and it's going to be on our group number. So if we click on the drop-down, you can order the group numbers by ascending or descending order. We're just going to go ahead and select ascending here. Now we have our group numbers in ascending order. If we scroll down to group 4, we see that every single plate in that group had 0 colonies. This is somewhat of a red flag. It could mean that there was a protocol issue, and there are various issues that can occur. For example, this could be a mistake in the transformation mix or a bad batch of competent cells. Because the entire group failed, it's likely due to a technical error, and we'll go ahead and remove all the data points from group 4 from our analysis. Now that we removed all the zeros and the empty rows, we're going to go ahead and take off our filter. So we're going to navigate back up to total number colonies and click on that drop down button, and we'll select clear filter. We'll insert a pivot table. Go to the Insert tab and click on Pivot Table. A new window will pop up that asks what data you want to analyze. We're okay with analyzing our data from this response form. And then it'll ask where you want to place the pivot table. We're going to place it in a new worksheet. A new sheet will open up with a pivot table field. If you go to the right-hand side, here are all the different field names, which is our column names. And there are 4 panels below that called filter, columns, rows, and values. These will be really handy when you want to organize your data in a very specific way. Let's walk through the basics of something you might actually be asked to set up in the lab. We'll organize the data by transformation type and calculate a few key values. We'll get the average total colonies, the standard deviation of the total colonies, the average percent red colonies, and the standard deviation of the percent red colonies. To do this, we're going to make sure that our cursor is within this pivot table field. Let's drag transformation type down to the rows panel. And now you can see our different transformation types here. We'll then drag our total colonies into our values panel. One thing to keep in mind is how Excel is summarizing your data. Here we see that we're getting the sum of our total number of colonies. However, if you right-click or command-click, there are different ways that you can summarize your data by. If you go to field settings down here, we can see that there are different ways such as sum, count, average, max, min, product. So double check to make sure that Excel is summarizing your data in the way that you would like it to. In our case, we would like to switch this sum of total number of colonies to the average. We will then drag our total number of colonies into this values panel. When you drag fields into this value panel, Excel might automatically set them to sum or count. That's why it's important to double check the summary type. Here we see that it is summarizing our data by the sum. However, if you right click or press command click, there's an option to go to this field settings. If we open up this new window, we can summarize our data by sum, count, average, max, min, product. So there's all of these ways that you can experiment with how you want your data summarized. For this case, we're going to go ahead and select average. We also want the standard deviation of our total number of colonies. So we're going to drag our total number of colonies field name again into the values panel, and we're going to change how the data is being summarized to standard deviation. We'll add 2 more columns into our pivot table. We're going to add our average percent red colonies. So we're going to grab this percentage of red colonies and drag it into our values panel. We'll change how it's being summarized to average, and then we'll drag the same field name to the values panel again and change how it's being summarized to standard deviation. If we look at our pivot table, now we have it organized by the transformation type, as well as our columns, which is the average total number of colonies, the standard deviation total number of colonies, the average percent red colonies, and the standard deviation of the percent red colonies. That's it for setting up a pivot table. Feel free to experiment with the field names and panel placements to organize the data in a way that best fits the insight that you're looking for.